Hello, everyone. So today I'd like to continue with the book Ending Aging by Aubrey de Grey, and I'd like to talk about two categories of sense that I mentioned in the last video. And I'd like to talk about mitochondrial DNA in detail. And I'm from a science background, so I find this interesting. If you are not great in science, you may want to refresh that because what I'll be explaining to you will be a bit challenging to understand, but even better to actually get the book and read about it yourself. So the first sense category that Aubrey de Grey wants to tackle in his approach called um, senescence negligent engineered negligent senescence is mitochondrial DNA, and it's shown that they there's a mutation in the mitochondrial DNA, what happens is um, they have been shown to completely take over the cell. So in older patients, all the mitochondria was mutated, and there was no healthy mitochondria. It was also found that was a case in only 1% of all of the cells. So it wasn't something very common, but only 1% of the cells actually had it. And so, um, what will happen is that these mitochondrial mutants underwent a process called clonal expansion. And the reason is because they simply camouflaged the they were camouflaged from the lysosomes because they produced less free radicals, and so they didn't have, they were actually deletion mutants, so they didn't produce the electron transport chain machinery. So because of that, the lysosomes attack, the lysosomes kill those mitochondria that have defected machinery in their electron transport chain. But since the mutants had big deletions that didn't have any of those proteins, they were camouflaged from the lysosomes. And so they replicated again and again. And then the whole cell was made entirely of these mutant mitochondria, and none of them were healthy. And so this is a big problem because what happens is, by the way, how did these mitochondria even survive without the electron transport chain? Well, it turns out that they had overactive citric acid protein. So they underwent all the other processes to make ATP or energy, like glycolysis and um, pyruvate, right? Cycle, as well as the Krebs cycle or the citric acid cycle. They just didn't undergo electron transport chain and oxidative phosphorylation, but they still got their energy from the other processes that I just mentioned. So they were able to survive, and they took over the entire cell. Why is that a problem? If they don't make any free radicals, you may ask. Well, what happens is, in order for them to undergo glycolysis and all those other processes, they need NAD plus. So they make a lot of NADH from glycolysis, from the Krebs cycle, but they can't replace it. The electron transport chain is used so that the NADH can give its electron to the chain and regenerate NAD plus, which is needed to undergo glycolysis and Krebs cycle. So the question is, how can they sustain themselves in order to keep getting that energy? Otherwise, they would die. But no, they take over the entire cell. That 1% of the cells only have the mitochondrial mutants. And so what can happen in this scenario actually is that this NADH that accumulates, right? There's an imbalance between NADH and NAD plus. So the NADH donates its electron to a membrane protein called PMRS to regenerate that NAD plus so that it can keep on generating energy from glycolysis and from the Krebs cycle. Well, that's great. The mutants survive, and if they don't produce any free radicals, who cares? But there's a problem with that, right? Otherwise, okay, these 1% are bad, but they can just undergo apoptosis without spreading it to other cells. But what happens when that PMRS gets reduced, it gets that electron, it then reduces another electron acceptor, and in the process, it turns oxygen to superoxide. And that's a problem. So, because then superoxide gets reduced back to oxygen, and in the process oxidizes cholesterol. So you get LDL oxidized form. That's a problem because the LDL cholesterol can oxidize, can leave the cell and create havoc in other cells. It's called oxidative stress. And that's what leads to aging through this mitochondrial theory. So this is the first of the seven damages that occurs because what happens is this oxidized LDL cholesterol causes havoc by oxidizing other molecules and causing oxidizing stress and causing havoc in cell metabolism and cell functioning. Just imagine if it can go to the rest of the body, which is what happens here in that 1%. It's only 1%, but still, if it can cause havoc to other cells in the body, pretty much the entire organism, that's the problem. So the question becomes, how can we stop these mitochondrial mutants from taking over? And the solution was actually a very clever one. We can do something called allotropic expression of these mitochondrial mutants. So we can move so essentially we can move the mitochondrial genome to the bomb shelter of the nucleus. So the nucleus can now produce all of those proteins. First of all, that's good because the mitochondria is at a much higher risk of mutations. So if we move it to the nucleus, there's a much lower risk of producing mutations caused by free radicals. So that's one. And the reason being is because if you make it in the nucleus, what happens is because of the electron transport chain and because of all that electrons being passed, there's a higher chance of free radicals causing mutations. So they're a hotspot, reductive hotspot it's called. So by moving it to the nucleus, there's a less risk of mutations. So that's for one thing. But also, if you make the machinery in the nucleus, these proteins are then going to travel to the mitochondria of the cell. And if they're in the mitochondria, now they can be recognized by the lysosomes of the cell, and thus they will be degraded and they wouldn't accumulate in the cell like they did before. And so therefore, you would prevent the spread of this oxidative stress. So that's kind of a very, very nice approach of doing it. Now, of course, there's a lot of things to consider here, right? So this method will prevent the mitochondria from getting into that abnormal state through donating the electrons to PMRS, etc., oxidating cholesterol, and causing havoc in the rest of the body. So it will prevent this clonal expansion from occurring. And so it will prevent this 1%, but even if you have this 1%, it will prevent it from causing further havoc. So you can actually eliminate the damage and fix the accumulated damage, in this case, the mitochondrial mutations, right at the source. So the way to do it is, as I mentioned, allotropic expression, so you express these proteins, which the electron transport chain machinery, which will then go to the mitochondria. Another way, because of the issues of sometimes passing the proteins through, there's a complex called the Tim-Tom complex at the mitochondria, and there's sometimes issues of actually moving things inside. And so what you can do is you can use also another approach is, instead of you using these proteins from humans, right, you're going to modify them. You're going to modify these proteins a bit so that they're made in the nucleus, but they're able to fold in the right way such that they can enter and be imported into the mitochondria. So you can change the amino acid sequence. And so this is all great, but sometimes the proteins are just in the water. They become too, because they're hydrophobic. They kind of curl in the water, and they're difficult to pass through the Tim-Tom complex or the mitochondria. So another approach. So one is to simply change the amino acid structure so that they're able to be imported by tweaking them, and research was shown from another organisms that have both. I think it was in legumes that have both the nuclear and the mitochondrial version of these mitochondrial proteins, and they saw the differences between the two, and they're just a few amino acids different. These are the amino acids that allow the proteins to be imported to the mitochondria. So you can simply change the amino acid structure by tweaking it a bit. Another way that you can do it is by simply borrowing these proteins from lower organisms like yeast, and therefore they're less hydrophobic, so they'll be easier to pass through the mitochondrial membrane through that Tim-Tom complex. Another thing you can do is, instead of being imported to the mitochondria, you can just do the whole stretch of these genes, the whole complement, so it doesn't have to go through, and so you're going to express all of these genes in one stretch, and so that's another approach. So it doesn't have to integrate anymore with the nucleus. That's an interesting one. I think that the more reasonable ones would be to either borrow it from lower organisms, or to change the amino acid sequence a bit so that they are able to be properly folded in a way that they can be imported into the mitochondria. And if we can get those proteins into the mitochondria, they won't be destroyed by lysosomes, and therefore the clonal expansion will not occur, and therefore oxidative stress will not be spread throughout the body. So this is really important to understand. The second approach is via intracellular junk, and this involves the lysosomes, which are those cell organelles that degrade cellular constituents that overaccumulate in the body. So once things stop working anymore, they don't serve their function, the lysosomes simply break them down, or when things become literally toxic to your body, the lysosomes can break those things down. Problem being is, if these proteins accumulate and they can be broken down, they take over the other proper constituents of the cell's functions. So they don't allow the cell to function properly. It's like they starve the cell if the waste over accumulates. It's like if you have waste in a city, everything is going to smell so bad, and that's why the life expectancy is longer nowadays, because we have much better sanitation system and waste management systems. So it's very important to be able to remove all that garbage from our cells, and that's the function of lysosomes. Problem being is, as we age, the lysosomes don't work as well, because there are things called lipofusins, and these are like aggregates within the cell that essentially they prevent the lysosomes from working, because they leave less room for other stuff and other enzymes in the lysosome to work properly. So they take up space and also dilute the acidity which is needed for the enzymes to work properly. And so the way to tackle that is to use enzymes from microbes, and these enzymes from microbes are known to digest in graveyards the dead remains of organisms into compost. So we can use these kind of like enzymes, proteins from bacteria or from yeast or fungi or what have you, to degrade these lipofusins to prevent to remove all that garbage as we age, so that the cells can function properly. This is extremely important in cells that don't divide, like in our heart and in our brain, because as you divide, their lipofusins content goes down and down. It's like a parent cell and a daughter cell, so it's been cut in half as it replicates. But because heart and brain cells don't replicate as much, the lipofusins content accumulate more in those cells, and these are very critical organs in our body. So using those bacteria or yeast or those microbes, because we don't have any enzymes in our body that can degrade these lipofusins as they age, because they're very non-degradable. But the yeast and bacteria in the graveyards that can degrade dead bodies are very, very powerful enzymes, and they will be able to break down those lipofusins, as well as other proteins that aggregates that are implicated in other diseases, like for example atherosclerosis, plaques from, again, cholesterol, oxidized cholesterol, in heart disease, or protein accumulation in neurodegenerative diseases like beta amyloid plaque, tau proteins, etc., as well as in macular degeneration for vision. For example, the A2E protein that accumulates in the retina and prevents you from seeing well, or that can cause blindness. So by using these proteins, from enzymes, we can break all of those aggregates, not only lipofusins because of aging, but also we can treat all those other diseases like stroke. We can prevent heart attacks, and also we can treat macular degeneration. And so there was one study which actually did show that it helped when you target the cells, the microbial enzymes were actually able to break down the lipofusins, which is a great result. Now we need to be able to first target the right cells, and then not only from that, we should be able to get it into the lysosomes because the lysosomes have the acidity for these enzymes to function properly. So we need to be able to inject either through enzyme therapy or through gene therapy, which is a safer approach, to inject these enzymes into the cells and then through the lysosomes. So, and there's a lot of, so we have to tackle all of these hurdles after, of course, we identify exactly what these enzymes are, so that we can inject them or apply gene therapy. Another hurdle is the immune system reaction, and I think gene therapy is a bit of a safer approach to prevent immune response reactions, but we can also inject it into the bone marrow where it's more safe as well. There's less of an immune response, but I think this problem will get solved because we're doing much better with like organ transplants and all that. So the immune response is probably least importance compared to actually getting it to the right cells, which we can do through a targeting stretch of amino acids, as well as getting it through the lysosomes, which we can do through sugars like mannose-6 phosphate, as well as through something called chaperone-mediated autophagy, which is a process in our own cells that we can get proteins into the lysosomes. So this is really cool. We can use these microbial enzymes to eliminate all the junk, all the garbage that we have in our body, in both our lysosomes and outside of our lysosomes, in the cell body or in the main part of the cell, to remove all that junk so that the cells can actually function properly, especially in critical organs like in the brain and in the heart. Thank you very much, guys, and I'll see you in the next lesson.